Yeah, dear. Very good. You got the number right too. Ton of different viruses. And how do I know it's not the flu? You see that? Any ideas? Yeah, so fever and joint pain or whole body symptoms more indicative of influenza. So that was fun. All right. So what we're up to right now is responses. So I want you all to move up to page 50. Okay. I'm going to be asking a ton of you questions, and like I said, the key thing is answer my question if I ask you, especially online students. And if your mic doesn't work, this is your chance to comment it, so I don't ask you. Okay? Because I hate awkward silences, and I won't pick on you more if you don't answer. Okay, cool. Now, can I get you to read stop right there? Okay, adaptations for entry and transmission. Now, like I said, the way they ask this dot point is they'll literally throw it to you as an exam question, very directly. The way I would have this categorized in your head is a different pathogens and what they do to enter and spread. Those are two things you need to understand. Okay? We explained prion disease last week. Who remembers what the entry or adaptations of prions were? Kirtana? Any ideas? Kirtana, what are prions? Oh, good. Quickly take a seat. What are prions? Prions are not. Okay. When you guys get asked a question, I don't mind if you're right or wrong. I want you to give me a first principle answer, right? So show me your logical explanation first. I care about that much more. Ethan, what's a prion? Come on, you're thinking too fancy. What is a prion? Martin? That's it, right? Prion protein is a misfolded protein, okay? Now, how can it be transmitted? So, Jessica, what are some different ways that prions can be transmitted?
It can be transmitted through surgical equipment, like indirect transmission.
Okay, so these misfolded proteins can spread from surgical equipment. Now, Narayani, are you a new student this week?
Hi. Yeah, I have this trial lesson, and then I will be joining in ADX from the next lesson. So yeah.
Okay, no worries. Yeah. Okay, Narayani, have you done Mod 7?
We've started Mod 7 in school, so we are not at the part where you have reached now. Yep.
No worries. We'll catch up with you as well. But Narayani, can you tell me anything more about prions? So we've said they're misfolded proteins. They can spread through contaminated surgical equipment. That's very specific. Remember, when I ask you guys questions, you should categorize it in your head. Do you know any other way prion diseases can be transmitted between individuals?
Oh, can it be bodily liquids?
It could be. So the key point here is direct contact, which is what I'm trying to get at, right? So you can directly ingest infected prion proteins. So if you consume cattle with prion disease, then you can get a human version of that prion disease as well, right? So that's an example of that. There is body fluids. It is technically true. Does anyone know an example of body fluids causing prion transmission? Blood? Yeah, so it's part of like the whole cannibalism thing. But what I'm trying to get is human growth hormone implants or injections that individuals got. So in the early or late 1900s, getting human growth hormone is released from the brain, right? Getting that injected into you from a person who's infected with prion proteins enabled the prion disease to spread as well. Okay? Good. So what I'm trying to get at here is sometimes in prion disease, the prion starts in the gut, let's say in ingestion. Sometimes it starts in the bloodstream. Let's say if you got, transplantation of fluids or tissue with prion disease. My question is, how does it get to the brain? Does anyone remember? Yeah, so there's these, what we call autonomic nerves, right? The way I think about this, because nerves, neurology is way too complicated for us, autonomic nerves do automatic functions, right? What are some automatic functions you're doing right now, Georgina? Yeah, right, respiration. Interesting point, that's secreted in your brainstem. So if you have a tumor or you have increased pressure in the brain that compresses that part of the brainstem, you die, right? That's why the brainstem is so important. Now, what's another vital biological function you're doing automatically? Thinking is, more or less automatic, but you got to think about the more fundamental things that are happening, 70, 90 beats a minute. Your heart, right? That's also an automatic function. You're not controlling that, right? So autonomic nerves control those functions, and prions like to hitchhike on autonomic nerves to get to the brain. That's the adaptation you can remember. Virginia?
The screen was
Screen was not on. Is it on now?
yeah, it was blinking.
Oh, okay. So when I look at something. Oh, I see. All right. I think it's because if I move this part of that. No worries. Cool. So that's a key thing you need to know about prions. Now, Kitty, you mentioned lymphocytes, and I'm sure it's because you read the word lymphocyte on the TV, right? I want you to revise, okay? What are lymphocytes? Does anyone know? Any online students know what a lymphocyte is? It's a type of white blood cell. Okay? You need to simplify things in your exams. So the way I remember this is prions are so tiny, they're a protein, right? So the way they can protect themselves as well is they can move into cells. And when they're inside cells, the body's immune system can't defend or fight the prion. And so prions can infect or move inside white blood cells. That's another adaptation they have. So move inside white blood cells, travel up nerves. If you say those two things, you get full marks of prions. Let's move on from prions and let's talk about the second largest pathogen. Ludwig, do you have a question?
Yeah, just regarding how prions want to go, like, why do they want to go to the brain?
Well, how do prions cause disease, Ludwig? You remind the class.
Well, they induce misfolding of all the other prions and form long chains or
Good.
fibers.
And where are prion proteins normally found?
Nervous tissue.
Good.
Yeah,
Right?
okay.
It's not that they intentionally want to go to the brain, it's that it's kind of like evolution, right? These proteins have found ways to get to the brain where they cause disease, and that causes them to persist and spread, and the prion disease becomes more common. Does that kind of make sense? So you don't have any sentience to them, they don't want to go to the brain. It's just that they tend to go to the brain, which allows them to misfold other prion proteins. Okay, so it helps them with their function. Yeah. Good. Does anyone have any questions at all before we move on? All right, good. Let's talk about the second largest pathogen. Ramith, what's second smallest after a prion? What pathogen am I talking about here?
A virus?
Good, right? So when we talk about viruses and their adaptations, this is my trick to you, right? If you know how a virus causes disease, you know all the adaptations of a virus. So if you remember from when I explained the behavior of a virus, you technically know all its adaptations. So let's start off with this. What's the structure of a virus? Let me ask. Good, Ramit, give it a go.
It has a protein coat, which is envelopes.
Good. Protein coat, that's protecting genetic information.
Yeah, it can be like the genetic information can be RNA or DNA.
Good. What's on the envelope, Ramit?
There's like it's like protein ends, right?
Good. There's these receptors. Anyone know what these receptors do? Why would a virus have these proteins kind of like keys? That's how I want you to think about this. These are keys. Good, right? So the receptor will determine the cell that this virus infects. If it's influenza, what cell type do we infect? What cells line your throat in your upper respiratory tract? Year 11? Eugenie, you should know this, the different cell types. What are lining cells called? Epithelial, very good, right? So epithelial cells are infected with influenza. What about HIV? Because I keep coming back to HIV. That can be a case study of an infectious disease if they ask you for one. Sheena, what cells do HIV infect? It's a fancy L word that Keith and I use. Yeah, so white cells, right? Specifically, the master white cell, or the manager of the entire immune system, the CEO of the immune system, the T cell. Anyway, Ramith, do you have any questions? Your hand's still raised.
Yes, sir. Does it use like reverse transcriptase to get into those cells, or?
No, that's not to get into the cell. It's solely viral glycoprotein, which are these proteins that you see on the surface that will bind to a receptor in a cell and then will fuse and enter into the cell. That's the first step. So that's one adaptation for entry, right? But this is the other thing you have to consider, everyone. When they're talking about entry and transmission, they're not talking about cellular entry. They're talking about entry into a host, right? We'll talk about that shortly. Okay. So this is how the virus enters your cell. What does it do then to then spread? Okay. So it inserts its genetic information into the nucleus of the cell. Okay. And what's the purpose of that? I guess the way I would break it down is once it's into the cell, a virus has two goals. It wants to replicate its genetic material, and it wants to make viral proteins. That's those two things, right? Genetic material, proteins. Now we'll start with you. How does a virus copy its genetic material? Because remember, most viruses are RNA. What are they going to do? Is that a process, or did you name an enzyme just then? You mentioned an enzyme, right? If you end in A's, you're an enzyme. The way you should think about this is to replicate the genetic material, it needs to use host cell enzymes, right? It needs to use your helicase, your polymerase. Who's had COVID before? Yeah, so COVID was doing this inside you while you were chilling in bed, right? It was using your helicase, your polymerase, your ligase to make more COVID DNA and RNA. Now, remember, those enzymes that you have are all DNA enzymes. So how does a virus convert its RNA to DNA? This is where you can mention.
So I use reverse transcriptase to turn its RNA to.
Yeah, because think about what transcription is, everyone. Transcription is DNA to RNA. So obviously this viral enzyme called reverse transcriptase is going to do the exact opposite. It's going to convert the RNA of the virus into DNA. And now since you have viral DNA, we can use host cell enzymes to replicate viral DNA. And this allows viruses to proliferate. You're now starting to make copies of that virus inside a cell. What's the other thing we need? Think about the structure of a virus. Ramith mentioned its genetic material in a protein capsid that's sometimes enveloped. How will this virus make more protein? This is an exam question. This is a very fair question to ask in your exam. It's 2 marks. Outline the process by which a virus will produce its viral proteins. So let me ask Tashria, what would you say? How would this virus make more viral protein?
I'm not sure.
You actually do know the answer to this. You just need to link the points in your head. Especially if I asked you how you make your proteins, what would you say?
Transcription and translation.
Good. Now, what can a virus skip, given that it already has RNA? Ashvia?
Translation.
What is translation? This is what you have to ask yourselves. Transcription is converting DNA into RNA. Transcribe means to make a copy of something, right? Same language. If I ask you to transcribe the dictionary, you need to copy every single word and its definition, right? Word for word. That is what transcription means. So we're copying this DNA. What is translation, Ria? You're converting one language to another language, which is amino acids. What can a virus skip, everyone? Step 1 or step 2? Logically makes sense. It already has RNA. So all that needs to happen is our RNA sits in the ribosome, and what happens? You make, we get translation. That's it. Now, these two processes are going to build more virus. And eventually, when there's enough virus, it will burst out of the cell, destroying it in the process. So, these are features of the virus that enable its transmission. So we've hit this tick for this part, right? So in an exam, I would say a virus transmits itself by fusing its host cell receptors with cells entering and using the cell ribosomes and cell enzymes to replicate its genetic material and proteins for transmission. But how does it enter? What are some adaptations of a virus to enter into their host? Now let's zoom out and think about humans. What adaptations does a virus have to enter your body? Hint, it's with a structure. What did we say was optional in this diagram about a virus? Sheena? The envelope, right? Why would you want an envelope, Sheena? What is an envelope made out of? Lipids, because when it bursts out of the host cell, it takes that lipid layer with it, so it helps with moisture. Do you all agree? When would we want to have moisture or retain moisture for a virus? Yeah, exactly, right? So a lot of respiratory viruses are enveloped. They need that envelope because they need that moisture to survive long periods of time on surfaces and in the air to spread between individuals. So having a viral envelope is an adaptation for respiratory spread, right? It retains moisture and allows the virus to exist for a longer period of time. Now, when would you not want to have an envelope? What are some viruses that would not care to have an envelope? Think about types of spread.
Viruses that spread through body.
Possibly, but remember, bodily fluids are moist. So viruses that spread by bodily fluids, already moist environments, they may not care about having a lipid envelope as well. Okay? So yeah, what I'm trying to get at is, there are some types of transmission where the virus will need to pass through really concentrated toxic substances like stomach acid, in which any moisture is sucked away, right? And those viruses that cause disease through that mode of transmission should be able to survive even the lowest of moisture conditions. What I'm trying to get at here is fecal oral viruses are typically not enveloped, right? Because they don't need moisture. They can survive in the toxic environment of the hydrochloric acid, which has a pH 2 in your stomach. So another adaptation is not having an envelope. Non-enveloped viruses only have a capsid, and this tough protein capsid helps them survive in toxic conditions in the body. I wouldn't use the word toxic. Toxic is a very misused term in medicine. You can just say in varied conditions in the body, e.g. stomach acid, right? So the two adaptations we've spoken about, everyone, is the envelope of a virus which helps it retain moisture, and that's supportive in respiratory transmission. And there's protein capsid, which allows the virus to persist. Survive is not the right word because, like I told you, viruses kind of not even living, right? So to persist in strong acid conditions, e.g. fecal oral transmission. Good. Now, you might be wondering, oh, there's so many pathogens and there's so many adaptations I have to know for every pathogen. My tip is, the main two they'll ask you is bacteria and viruses. And if they ask you anything outside of that, it'll be max 2 marks. So just know one thing. In the trials, they have asked about prions, but they've never asked about protozoa and everything else. Okay? So that's why I taught you prions in a bit of detail. It's been asked before as a trial question. Good. Let's move on to bacteria. Bacteria is actually quite easy. If you remember the structure of bacteria, you will know the adaptations. What are some adaptations of bacteria? Sorry, a bit louder. How is that an adaptation? You're completely right with the structure. I'll give you that. That's the structure of a bacteria. You're completely right. Good. Could you repeat that? I think you combined 2 words together. Which? What's this called? Flagella, right? So what does a flagella help bacteria do? That's an adaptation. Yeah, so it enables motility, right? The flagella literally swings like a turbine, and the bacteria will travel in water, which is what 60% of you is made out of. So 1 mark, if you say flagella, enables motility and thus transmission within cells of an individual. Right? Lupin, you have a question?
Would another adaptation be the cilia?
Okay, another point. I know there's so many words in medicine, they all sound similar. Cilia is not a word we'd use for bacteria. Cilia refer to tiny hairs in our respiratory tract that are beating upwards. They beat upwards at like 60 times a minute, so every second they're beating upwards, right? That's in the human body. I think you're referring to these hairs that are on a bacteria. That is not cilia. What is it called? Good, pili.
Oh, yeah, that's right.
Flagella and pili. That's completely fine. Honestly, they're probably very similar in size, shape. The only thing is cilia are always beating upwards. These pili, they simply anchor on, right? They're kind of like rods that will stab into a cell to attach it so it remains on that area. What kind of, it's a hard question, but I want you to think laterally, right? What kind of bacteria would really rely on that pili? So think about, when would you, imagine you're a pathogen, when would you really need to hold on for dear life in the human body? One example is in the respiratory tract, because you said those cilia, or those hairs, are beating upwards to try to get these pathogens out. So a lot of respiratory pathogens would have pili for adhesion to cells. What are some other examples? Yeah, potentially. Potentially, but there's no direct active forces on the skin at rest. You have to use your hand and rub it off, right? And think about parts of the body where there is movement of fluid down a passage. Urinary tract, right? Even gastrointestinal pathogens, right? They need to survive against the peristalsis of the GI tract. But urinary tract pathogens, E. coli specifically, right? That's a bacteria that really uses its pili to anchor on into your urinary tract. So even when you are urinating, it can spread and cause infection. Good. So do you see how knowing the structure helps with all these other dot points? You don't need to memorize anything for bacteria, just know what it looks like. What's one more? What additional layer does a bacteria have that can make it? Pardon? Yeah. This cell membrane, I don't think it's made directly out of phospholipid. It's made of something slightly different. Yeah, you could say the cell wall the thickness of that protein cell wall can limit any immune cell activity directed against it. So it's kind of like a protection. But that's a bit equivocal. Any other ideas, anyone? Gayatri?
I'm just confused for this one. Can I say that produces toxins, which increases its like, yeah.
You could technically, you could say toxins enable, cell death and facilitate transmission. But do you all see how that's very vague? Most students can say that. So what they want here is to show that you understand the structure of behavior. So if you're going to say a toxin, you should probably name a toxin. Does anyone know any names? Any toxin names? I'll give you one that I remember. I actually had to study this very thing in last year's exams because it was very biomedicine based. I was learning what you guys are doing. Now, this is a fancy toxin, so you don't need to memorize the name. I'm only giving it to you because you mentioned toxin. Is this enzyme breaks down hyaluronic acid. Does anyone know what hyaluronic acid is? You might have seen it on the skin creams, supplements. It's basically, it's a substance found in joints and tissue. So this enzyme destroys that substance and enables a bacteria to spread in tissue. Right? So it's basically a toxin that causes tissue destruction. Okay? There's many others you can see how they've broken this down. Maybe you can remember this. There's endotoxins and exotoxins. This just refers to whether the toxin is killing inside cells or outside cells. Exo means outside. It's damaging the surrounding tissue, whereas endotoxins can enter into cells and damage them. Good. One really good toxin I would have wrote, this is actually pretty good, is IgA protease. Anyone know what that would do? Has anyone heard of Ig? If you've maybe gone and got a PCR test, you might have seen IgM, IgG. Has anyone seen that before? Okay. When you see the word Ig, those are antibodies. Does anyone know what antibodies are? Good, Lookman. Safia, what's an antibody?
Don't antibodies fight like any viruses or bacteria in your body?
Good. Antibodies are proteins we make to destroy these pathogens. So what do you think? So Ig means antibody. This is an IgA protease. What do you think it's doing to the antibody? Destroying it. Bacteria have developed toxins to destroy antibodies. Okay? IgA is a specific subtype of antibody. The way I remember this is IgA, it's kind of like little minefields in your respiratory tract. Your body has secreted IgA, which sits in the respiratory tract waiting. And when a cell sits in that IgA, it will bind to it and just help destroy that pathogen. It's kind of like a hunter's trap, but it's in your respiratory canal, right? These bacteria have now made toxins to destroy those antibodies. I'd say that'd probably be the best one to memorize if you were going to memorize a toxin. This is what I've said. All right, cool. Let's move on. You don't need to memorize all of this. This is a case study on Streptococcus pneumonia. Guess what disease it causes? Good. What is pneumonia? Lung infection. Infection of the alveoli. Okay. Good. You don't need to memorize any of that. Just have a read if you want to, because you can pick specific adaptations you like. Here's my little tip to you. Protozoa just copy bacteria, but be smart about it. What do protozoa not have that bacteria do have? Good. What's another feature about protozoa that's different to bacteria? Let me ask, Lookman, I know you'd know this, but let me ask Kashmir. What's the difference between protozoa and bacteria?
They're eukaryotic.
Okay, good. Eukaryotic, so they have a nucleus as well. So do not mention anything about a cell wall. So just copy pili, flagella, you'll get the marks of protozoa. Okay, good. You should probably know one example of a protozoal disease. Do we know one? Malaria. Very good. So what causes malaria though? That's the name of the disease.
Plasma.
Vector. Very good. Good answer. The Plasmodium protozoan. Very good. All right. Let's move on. I've never heard them ask about fungi. I'd say it's so low yield. Maybe just memorize one specific point about fungi. If I was to give you one, going through this, probably be cell wall. Okay? Cell wall component of fungi can protect them from attacks. The reason that's important, and you can kind of derive that from first principles, is if you know what fungi look like, you would have given me this adaptation. They have a cell wall made out of chitin. Chitin has proteins and sugar. Chitin has proteins and sugar. Can assist in survival. What's another thing? What do you all hear about fungi? When we talk about their reproduction, what word do we hear? Spore. What is a spore? We never really defined it. In your head, I'm sure you think of little balls, right, that are floating in the air. But a spore is kind of like a dormant form of that fungi. Okay? And since it's dormant, does it use as much energy as an active fungus? No. And it's also harder to kill. Fungal spores are much harder to kill than actual active fungi. So you can also talk about spores that are dormant forms of the virus, not virus, of fungi that can persist in adverse conditions, enabling spread. This enables their spread. That's it. Key points, everyone, focus on bacteria and viruses. You should know a lot of good adaptations for those two. Have one for every other pathogen. And I'm happy for us. I'll briefly talk about parasites too. It makes a lot of sense. Okay, so when we talk about parasites, Victoria, tell us about how we should think about parasites. Because technically, every single pathogen we've discussed is a parasite, right? Parasite is any organism that has an adverse or bad relationship with the host. Okay, we were technically parasites in the womb, and up until 18 years, we're financial parasites as well. Victoria, how does a parasite behave? What are the two different types when we talk about parasites in the HSC? Any ideas?
About the types of parasites?
Yeah, how do we think about parasites? You might have remembered the word endo and ectoparasites. Do you remember what they are?
Yeah, so endo is a parasite inside and then ecto is outside.
Good, that's right. And with endoparasites, what do they tend to look like? Here it is. We'll help you out. If it's inside the body as a parasite, it looks like this usually. Worms. What's a name? What's a fancy word I gave you all for worm parasites? Good. Helminths. Helminth. Helminths. Okay. Good. What about ectoparasites? What are some examples of those? Let me ask Raya. What are some examples of ectoparasites? Raya, ectoparasites. Tell me something about them. I'll come back to you just in the interest of time. Ramit, give it a go.
Would ticks be an example one?
Yeah, ticks, mites, lice. Keeps going, right? Ramit, what's the one thing ectoparasites want?
They want nutrients in blood, right?
Blood specifically. Very good, right? Now, let's start by talking about ectoparasites. Imagine you're an ectoparasite. You found the skin, you pricked your mouthpiece inside the skin, and you're obtaining blood. What would you want regarding the diameter of this blood vessel? Would you want it to be a big blood vessel or a tiny one? Yeah. Now, did you know when you get bitten by a mosquito, what do you notice in the skin after the mosquito's gone? Yeah, it's bumped, right? And what color is it? What does that tell you about the blood flow to that area if it became red after? Increased. Now, the reason that happens is because these ectoparasites will secrete chemicals that will cause the diameter of this blood vessel to become even larger. That causes, superficially, what you see in the skin as redness, a bit of swelling. Right? But does it make sense as to why the mosquito wants that? More blood flow for it, right? More nutrient intake. So that's an adaptation of these ectoparasites. They release chemicals that cause blood vessel dilation. What's the word we use for blood vessel dilation? Does anyone know? Because in Module 8, you're going to have to use this a lot. Yeah? Vasodilation. Yeah.
A leech.
Yeah, a leech would also be another example of an ectoparasite. Very good. Right? It's a macroparasite, meaning that you can see it with your eyes. It will do the same thing as well. Yeah. Really good. There's medical conditions where we actually use leeches. We'll talk about that in our break, but we actually use that in hospitals sometimes. Not any hospitals I've seen, but has been used. Okay, good. And increases blood vessel permeability as well. Just a brief point about blood vessel permeability. What symptom would you notice if there's increased permeability of blood vessels? Well, think about it. What's in blood? Blood is fluid and red blood cells, and a few other things. We'll forget about that for now. If you increase the permeability of blood vessels, aka you increase the gap between cells on the blood vessel wall, what starts to happen? Things can leak out, right? And so you'll notice that the skin becomes bumped or swollen. So swelling is caused by increased blood vessel permeability. Redness and heat is caused by the blood vessel dilating. You might be wondering, well, why are we going into so much nitty-gritty detail on this? It's because you're going to learn this in much more detail in the next inquiry question, which is how your body responds to pathogens. This whole thing about vasodilation and blood vessel permeability. Good. Any questions at all? Rabbit, do you have a question or?
Yeah. So just a quick question.
Yeah.
With like ticks and stuff, they have like a hooked
Mouth part?
yeah, that like
Yeah.
a lead that allows them to Is that like another adaptation?
Yeah, definitely. That will get you a mark as well. It's a curved mouthpiece that enables anchoring into body tissue to obtain more nutrients. So there's a lot to it. I honestly think there's hundreds of adaptations for all these guys. So you have to think about them. How long have we been around? Any ideas? According to the archaeological or fossil records, like 3.5 to 2 million years or so, right? Now, these pathogens have evolved over that same time period, as we have as well. So they have so many adaptations that allow them to infect and cause death in humans and other animals. Okay? If you start thinking about it, why does COVID spread via the respiratory route, everyone? It's very easy to spread, right? And why does it cause coughing and sneezing? Because it enables it to spread by the respiratory route. We're all just serving kind of like vehicles for these pathogens. It's quite interesting. Cool. Any questions so far? This takes us to the next inquiry question. Responses to pathogens. I think this was a dot point that I did in the HSC which made me realize how insanely complex we are in terms of the human body, right? You could spend your whole life studying biology or medicine, and you'll only probably get a drop of the entire ocean, which is what you are, what processes you engage in. What we're doing now, it's going to be challenging for you, but scratching the surface. This is the kindergarten stuff of how the human body works, which is exciting and interesting because our body is much smarter than we could ever be. Now, with this inquiry question, Sashita, could you just read it out for us? Specifically, not the dot point, yeah, just the inquiry question.
How does the plant animal respond to infection?
Okay, cool. Now, Sashita, why should you care about how plants respond to infection, right? Animals are interesting. It's immune response. I'm gonna write that right here. But is there any use for learning about plants? Well, for most of you, no, right? But to make it very simple, the reason you're learning about plants is because plants are a simplified life form, right? You have to think about this. In science, when we do experiments, before we start human trials, what do we work on first? Smaller animals, like mice, right? Simpler models, which we can study, change the life expectancy of, etc. Plants are a simplified model of humans. That's how I think about it. Whatever's happening in a plant, the fundamentals are also happening in a human. And this also applies for the immune response. Plants have an immune response. You'd be surprised because they get infected. You all learned about bacteria, fungi, viruses, right? Plants respond to them, so they have an immune response. So studying how these plants respond will give you the ABCs to understand how you respond. Could I get Martin, could you read the first dot point out? Actually, without reading it, Martin, tell me what the most important parts of this dot point are when you're ready. What would you underline that students must know or must not miss? Good. So that's, yeah, for example, you only need to know one or the other. You need to memorize both. But the tip I'll give you is this part. This sucks. You need to know a native Australian plant. Like I said, biology is much less memorization now. I promise you, when you sit your exam, you'll be wondering, how did I learn all this info and only apply about 30% of it in that exam? Right? That's how it tends to feel when most students finish their exam. One of the few things you need to memorize is listed here. You must know an Australian plant and how it responds to a specific kind of pathogen. Now, coming back to you, Raya, which pathogen is more common? Fungi or viruses? Which one should we go with? Fungi or viruses? Safia, I'll let you save.
Oh.
Oh, Raya, you've got it? Thanks, Raya.
Fungi?
Sorry, I thought your mic wasn't working. Yeah, give it a
No,
go.
sorry, you keep asking me questions when I'm away from my computer.
No worries. Yeah, that's all right.
Fungi?
Good, fungi more common, right? So what I would recommend is just knowing fungi, one fungi, and how a plant responds to that fungi. Okay? The example I'll give you is eucalyptus. And the example of the fungi I'll give you is Yersinia. That's the genus of the fungi. There's many different kinds of this. And this causes rust. That's a disease it causes. Jot it down. One of the few things you'll have to memorize. That's it. All right. Good. Any questions so far? Rabbit, question?
Yeah, hey, so is that the same as like wheat rust? Like the same path?
Yeah, it's the same. This fungus also causes wheat rust as well. This is just eucalyptus rust, which is just rust in a different plant.
Oh, okay.
Same type of disease.
Actually,
Look, question?
yeah, this is just out of interest. I noticed that like when I'm outside, like 95% of the plants I look at, they're all infected. Like
Yeah. Well, it's
yeah,
good
like
it's good that you pick that up, right? I think a lot of you, like what we're learning right now is just pen and paper stuff. But like, I see the human part of stuff, right? I see patients with end-stage cancer, sepsis, infection, but you also see a lot of infection as well. When you look around you in the environment, a lot of plants. I think most plants have infection. That's definitely true. That tells you about the quality of the plant immune response. It's not as powerful as a human or an animal one, but you're right, most of them are infected.
So would that be the reason why it's so prevalent among them?
I wouldn't say it's because the plant immune response is just weaker. That's one part of it, but there's many other factors. Like viruses have a lot of pathogenicity, which means adaptations for entry and transmission, etc. Good. Question?
So you know there's like some, for example, the eucalyptus plant is infected,
Yeah.
and an animal consumes it.
Yeah.
Be passed on.
No, so that's the thing, right? Like you have to think about this. These pathogens, they have to choose one game to play. Infecting plants or infecting humans. They're completely different games, right? It's kind of like getting a sprinter and getting to run a marathon. Would he be able to do a successful marathon? Maybe not. Okay? It's the same principle here. These plant viruses, they have adaptations to penetrate a cell wall, or they have adaptations to survive against tree bark and gum. When they see your white cells, they will get destroyed. There's no way they cause infection. So I'd say all plant infections cannot cause animal infections. So you can eat, it's more the decay, right? There'll be a lot of bacteria on decaying vegetables and such, which will cause human disease. All right, cool. Now, for the immune response, let's talk about what that is. Does anyone know how you define an immune response? It's a vague term. Immune response is just any coordinated action to a pathogen, right? Did I say the word immune cells by definition? No, because you don't need immune cells to have an immune response. We have immune cells. I'll tell you right now, plants do not have immune cells. They still have an immune response. Their normal plant cells can engage in an immune response. Okay, so when we talk about the immune response, this is how you need to think about this. With plants and animals, it's the same thing. We have physical and chemical barriers. This is even before the immune response. Right? Kind of like with a castle, how you have that fort, or you have like a lake with alligators surround it, right? To not let anyone enter. That's the first barrier, right? It's your first line of defense. Your second and third line of defense are typically your actual immune response. Now with plants, they have mainly two lines of defense. So they have their first line, which is their physical and chemical barriers, and their second line of defense is their active immunity. Plants lack this third line of defense, which humans have. So we're not going to talk about it just yet, but that is a secret as to why the human immune system is much more complex, has cells, and actually remembers diseases, right? Do you realize that's how vaccinations work? The reason you're injected at 3, 6, 9, and 12 months as a baby with measles, mumps, rubella is because 17 years or 18 years later, your body still remembers being injected with that pathogen. So when it sees it, maybe next year, if you get infected with mumps or measles, it remembers what that pathogen looks like, and it knows to destroy it before it even causes disease, right? Plants can't do that. That's the third line of defense. But does that make sense to everyone? We have 3 lines of defense normally. Plants have a first and second. They lack a third. The first line of defense is not the immune response. Do you all see that? These are barriers that just exist. They're always active. They're always there. This barrier blocks against entry. Okay? Now let's talk about the plant. What would that first line of defense be? These 2 diagrams to kind of help you out. I'm sure you all saw this in year 11. What are the barriers that we see here? Physical or chemical?
A waxy cuticle.
Good. Good. So thick, waxy cuticle. What else does that do, Pranav? Because you actually need to know this in Module 8. What's the other function of the thick, waxy cuticle? Any idea? What else happens from the surface of a leaf? You learned this in year 11. Underside of the leaf, what else happens? Water? You mentioned a substance. I'm asking for a process. Yes. What do you call evaporation of water from the underside of a leaf?
Stomate regulation.
Transpiration. Did you all learn transpiration? Come on. You guys just control out bleeding. Photosynthesis. Okay, so the thick waxy cuticle prevents against water loss. That's actually something you need to memorize for Module 8, which is homeostasis or balance in plants. That's why I brought that up. Really good. Thick waxy cuticle, that's one key physical barrier. So in your notes, you should be writing physical, chemical barriers. It's also in your notes. But one physical barrier is a thick waxy cuticle. If you'd like, just highlight it right there. What's another physical barrier? There's a lot here. What can the stoma do? Yeah, very good. So open and close to allow gaseous exchange, but in infection, what will the stoma do? They'll close. So stomatal closure, that's technically an active response. So I would not directly include that as your physical barrier, but I'd say I'd leave that for now. We'll come back to stomatal closure because that's a process. You'll see that's a process that needs to happen. It's a response. I'm asking you what structures are here. So what else do we have? So that's up here. What about here? Yeah, right? Bark is one of the simplest defenses for a plant against infection. So tree bark and waxy cuticle. Done. Let's move on from physical barriers. What about chemical barriers? Any ideas, anyone? If you do chemistry, you might know this. Have you studied the soap tree? So indigenous uses of acids and bases. Do you remember studying that? What's an example of a base that's used?
Psychosin?
Very good. Psychosin is a basic toxin. Yeah, look, I think you could use that. I just don't know if that toxin is a toxin against pathogens. I think that's more a toxin against consumption. Yeah, the animals. So psychacin is found in the seeds. For everyone wondering what that is, cycads have psychacin, which is a toxin in the seeds, but it's not really a defense against pathogens per se, which are much smaller, right? I would say the example I'd use is saponins. They come from specific plants like the soap tree. You don't need to memorize this, but this just helps you understand what saponins are. It's like soap. Plants make their own soap to destroy and prevent entry of pathogens. So some of these plants will release saponin, which is a basic substance, as a chemical defense against pathogens. Let's think about the human equivalent here. We've been talking a lot about plants. What's the human equivalent of physical and chemical barriers? Very briefly. Kirthna, what do you see that's a physical barrier with humans? Just look at yourself. What do you notice? Skin, right? Does anyone know, are these skin cells alive or dead? They're dead. Skin's actually thick. The bottom cells of skin are alive, and when they move up, they die. Why would they die? No cell, no infection. Does that make sense? That's the logic. Cells die so that they can't be infected. And for an organism to cause skin infection, it needs to travel low to the bottom layer of that skin, which is much, much harder. It needs to penetrate through all that keratin and protein. Okay? That's why skin infections are not common unless you have a cut. Cuts get infected because that's a break in the skin. Does that make sense? Good. What's a chemical defense that we have against pathogens? The idea is chemical. Very good. Stomach acid's a very powerful defense. Ramit, anything else?
Would like mucus be an example of one?
Mucus is more a physical one. It does have chemicals in its structure,
Hmm.
but it's much more a physical barrier because it will immobilize the pathogen and allow that cilia in your respiratory tract to kick it up. Question?
This is like completely off topic.
It's fine.
But
Is it science?
It's science.
Yeah.
You know how you said like skin cells are dead?
Yeah.
Then how does like eczema work?
Very good question. To simplify things, eczema is called dermatitis. That's the scientific name. That's due to mutations. It's not really even inherited. Sometimes it just happens randomly in people, but you lose water in the skin. And when you lose water in the entire skin layer, the skin becomes dehydrated, it breaks. There's risk of infection because now it's compromised, and that's making it redness, swelling, superimposed infections, etc. Does that make sense?
Yeah.
It's a water problem. Your skin's not retaining the water. So it becomes dry and also infected. Good. All right, let's move on.
Excuse me, sir.
Yeah.
Would antioxidants be one?
So we need to think about this. Antioxidants protect against oxidative damage, right? Oxidative damage is usually what our body makes all the time. Think about this. When we eat food, we have metabolism, which is chemical reactions. Chemical reactions create waste. Oxidants are a type of waste. So our body counteracts that by making antioxidants, some which are vitamins and minerals. A lot of vitamins and minerals, think vitamin E is an antioxidant, right? So I wouldn't say that's a defense against pathogens, but that's in the world of preventing cancer, healing cellular repair, antioxidants.
Okay.
But there is a role of oxidants here, and we'll talk about that shortly. All right, let's move on. So any questions about physical chemical barriers? Good. Let's move on to our active defense. Now, my question is, how do plants, how does any living organism know when there is an infection or a pathogen?
Antigens?
Imagine if we had to find that out ourselves using our brain, looking at our skin every day, looking for any signs of pathogen or infection, we would utterly fail, right? So my question is, how does your body already know when a pathogen has entered and to start fighting and attacking you? So look, when you've said the A word, could you repeat it and tell us what an antigen is?
So an antigen is just marker molecules on cells, and basically the body just recognizes that it's foreign.
Good. Marker molecules, right? One thing I would say though, Lukman, is that antigens don't have to be foreign. Antigens are any molecule. Give me an example of a molecule, Pranav. Yeah, literally any. Yeah, that's an example. It could be an antigen, right? Sugars, carbohydrates, proteins, fats. Those are all antigens. Does that make sense? Any molecule is an antigen. When we talk about antigens, we can separate it into two categories. Self-antigens, these are yours, right? So these are your proteins, your fats, your carbs. The way you think about this is it's kind of like a pencil case, right? Some of you have written all over your pencil cases or put stickers or your own branding, which signifies it's self, right? Whereas everything outside of that is other. The same thing with antigens. Look at your body. Everything that makes up your body right now, those are self-antigens. All the molecules, the atoms, those are self-antigens. Everything outside your body in the observable universe is a foreign antigen. Does that make sense? A shard from this table, if it stabbed you, is that a self or foreign antigen? Foreign, right? If that tissue paper got in your eye and irritated the eye, is that a self or a foreign antigen? Foreign. So now we're getting to this understanding that antigens are just any molecule, and they can be self or foreign. Now, what does the body do to foreign antigens? What does it do? Mounts a immune response. It doesn't even have to be a pathogen. Anything that is foreign will will have an immune response mounted against it. Give you an example. Does anyone know any diabetics? Any type 1 diabetics at school? No? Some of them use an insulin pump. They have a needle in their stomach, their abdomen, that will release insulin. It's via a program as well. Now, that needle has an immune response mounted to it over time. The body starts to form adhesions and wall off that needle because it thinks it's a foreign object, which it is. And so it rapidly mounts an immune response. So that's just an example to show you that it doesn't even have to be a pathogen. Anything has an immune response mounted against it that's foreign. What about joint implants? What do you all think? They're foreign. They will have an immune response mounted against them. That's why we have to use titanium or stainless steel, because that survives even despite all these chemicals the body will use to try to destroy it. Good. Do you have any questions about antigens? Okay. The reason I stress this so much is this is a crux of the human immune system as well. You don't know what an antigen is? That's not good, because that's your ABCs. And students often confuse antigens and antibodies. The way I remember it is antigens generate an immune response. Antigens generate an immune response. Okay? Antibodies are what we make to bind to and immobilize these antigens. Question? Lookman question?
So when it comes to pathogens specifically, is that when antibodies bind to their antigens and like immobilize them?
That's exactly right.
Yeah.
Exactly right. So pathogens are an example of parasites that have foreign antigens, and so your body will produce antibodies that will bind and lock it up. The way I think of antibodies, analogies help because this is all very complex stuff. Antibodies are like handcuffs. They'll bind to things and immobilize them. Do handcuffs kill people? No. Antibodies do not kill pathogens. They simply immobilize or neutralize the pathogen. Okay? Ramith's question?
Yes, sir. So with anaphylaxis, does it misinterpret foreign antigens?
Yeah, anaphylaxis is very interesting. Does anyone have anaphylaxis? Anyone have asthma, eczema? No? Okay, interesting. Well, anaphylaxis is one of those very severe life-threatening conditions where your body responds to a foreign antigen very inappropriately. So it could be eggshell, it could be eggs, it could be shellfish, it could be peanuts. What happens is the body responds to it by thinking, it's a foreign antigen, it needs to mount a significant immune response. And we'll talk about what the immune response is because you don't understand that yet, but that can lead to death if it's unregulated. So an overly powerful immune response. So yeah, you're right. Does anyone know what the treatment is? Your friend starts telling you their throat is swelling up. EpiPen, right? Adrenaline. Adrenaline, you'll understand this shortly. What adrenaline does is it constricts blood vessels. And you'll understand then that the immune response involves the dilation of blood vessels. That's why we use adrenaline to constrict it and reverse it. So what you're learning now is what we use to save lives. Cool. All right. Now, with active defenses, remember, this is once a pathogen has already entered into a plant, right? So now it's the immune response that's being mounted. You now know this plant is looking for foreign antigens. When it finds these foreign antigens, it will mount its active defenses. So mounted, when exposed to foreign antigens. Now, I'll give you all a tip. What I've written here in these notes is too much for you to know. Don't worry about that. Don't worry about effector-triggered immunity, pattern-triggered immunity. That's all just very extra stuff. What I want you to just remember it as foreign antigens, plant will mount an immune response. Okay? This is just the details you can see here on how it will identify these foreign antigens. But again, out of the syllabus. I wouldn't waste your time learning that because there's so much you need to know from biology. Oops, sorry. I think that's my alarm for you all to have a break. Okay, let me just quickly finish active immunity and then we'll take a break. Okay, cool. So once the plant recognizes a foreign antigen, it starts engaging in a couple of responses. You can see here. I would say you should probably remember these 3 responses. They make a lot of sense. One of the first things that the plant will do is it will start producing free radicals. So let's draw this out so we understand this. Plant cell has now been infected. Okay? What the plant will start doing is, number 1, producing free radicals. Narayani, do you have any idea what a free radical is? Sorry, I think you have a question. You can ask me a question first.
So my question was for antigens and antibodies. So what's the main difference between antigen and antibodies?
Good question. So the first difference is to do with what their purpose is, right? An antigen is any molecule in the observable universe, right? And we just break it into the molecules that make you up, which are self-antigens, and molecules regarding the rest of the universe, everything outside of you, which are foreign antigens. Antibodies are proteins. That's the first key difference. There's one specific type of molecule, whereas antigens can be anything. The other key difference is that antibodies are proteins that we make through our immune system to fight and destroy any foreign antigens. Let's say that your thumb gets pricked with a little wood piece. Foreign antigen. Your body will mount an immune response. It will produce these antibody proteins. We're making this hand gestures because they look like wire-shaped molecules. That's what an antibody looks like what my hand is doing here. It'll go and bind to and immobilize the antigens in that wood piece to help destroy it. Does that make sense?
Yep. So antigen is basically any object or like any molecule that is
Molecule. Yeah.
Okay.
Right? Yeah.
Yeah. Yep.
Good. And the way you split it is self and foreign. That's the key thing.
Okay.
Good. Yep, Hashira, question? It's really good. I like it. We have a lot of questions this week, which I'm very happy with, because it shows that you're thinking hard.
So I think I was supposed to answer a question. I forgot to lower my hand. Sorry.
Oh, no worries. Maybe you can help with this. What is a free radical, Hashira? Any ideas? Free radical.
I'm sure. I'm pretty sure like it ruins the purpose of something, but I forgot what it specifically does.
Do you remember which module you learned it?
Oh, it was mod 5?
Close, mod 6, right? You learnt it regarding mutagens. Very good. What is a free radical, Pranav? Can you remind the class? In loud.
They damage the DNA.
Radicals rip electrons off things. That's the key thing you need to remember. Another name for a radical in chemistry is oxidant. Right? If you do chemistry, you should know an oxidant causes another substance to oxidize or lose electrons, aka ripping electrons off things. Okay? When you rip electrons off things like DNA, you cause DNA damage. What does that do to a pathogen, everyone? Think about this. Imagine a bacteria has infected this plant. The plant has started making free radicals. What is that going to do to the bacteria's DNA? It's going to rip electrons off, cause mutation, DNA damage, and kill the bacteria. What's the side effect though? Can you direct free radicals only at one's pathogen? Crossfire. Innocent civilians or plant cells will also die in this process. Does that make sense? Good. E.g., pathogen DNA. What's another thing that a plant would do? Well, it's going to reinforce its cell wall. It's going to start making more cellulose to prevent any further pathogen entering. That's a second key difference. Start drawing this out so you can really appreciate what's happening. Right? Second thing is cell wall apposition. This stuff is visual. Wall apposition. Make more cell wall. And the final thing, what did we say was a physical barrier, but I told you it was more of a response? Opening and closing stoma. So the last difference is stomatal closure. More of a response, so we put it here. The plant will close its stoma to minimize any further pathogens entering. Good. Questions? I will start with Lukman. What question do you have?
Sorry, just going back to the antigens. So do you know how?
Lukman, not allowed to say sorry with questions. Just
Oh,
answer
okay.
them,
Going back to the antigens. Say if a wood piece gets, like the analogy you referred to before, say if a wood piece got stuck in our finger, do you know how for pathogens, antibodies bind to their antigens, but what would antibodies bind to? Would it just stick to its surface, or?
Yeah, you literally start producing antibodies that will start binding to the surface of that wood chip. Yeah.
Oh, okay.
At microscopic level. Remember, this is all you wouldn't see these antibodies. But
So it's not like that wood has any like projection, like
But also think about this: is wood sterile? Everyone, is it pure? Is it clean? No, it probably has bacteria, viruses, and so you start making antibodies to that as well.
Okay.
Right. So bacteria on the surface of the wood. Yeah. Good. There was a question. Narayani, did you have a question as well?
No, I just forgot to put the thing down.
Raman?
Yes, sir. So for each specific antigen, there's a specific antibody.
Very good. I haven't come to that yet, but he's actually mentioned a very good point. Did everyone hear that? So for every specific antigen or molecule in this observable universe, there is a different antibody that can be made towards it. And just try to understand that. So what that means is every time you fight a different virus or a bacteria, your body produces a different antibody, right? And the antibodies come from cells, right? So to have a different kind of antibody, you need a slightly different kind of white blood cell, right? Because white blood cells make antibodies, and a slightly different version of a white blood cell will make a slightly different antibody. Does that make sense? So let's say next year there's a new pandemic. Say it's not COVID, let's say it's, I don't know, some weird new respiratory disease. Let's say Ebola 2.0. Right? That's a new disease your body has never seen, everyone. Okay? What must your immune system do? It needs to create a specific antibody to fight this new pathogen. Where does that antibody come from? A specific white blood cell that was already in your body this whole time. Just think about that. You right now, you have a white blood cell that can fight. So one white blood cell for every single different kind of infection, including infections you've never even seen before. That is how complex your immune system is. I found that one of the craziest facts. You were born with a white cell that was born to fight COVID-19, right? It just never activated until 2019. Okay, so maybe I'll explain how that works, but it's quite interesting. Cool, all right, take your break now. You go outside, stretch, get some air, and we'll recap at about 12:24, okay? Okay, I think we have questions. We'll just wait for sound to kind of die down a little bit, and then I'll answer all your questions. Nice. Okay, questions? Let's start with Ashita. What questions do you have?
So this isn't related to the module we're doing right now, but I have a biology assessment coming up this Friday, and it's about this and mitosis. Would you be able to explain the two processes really quickly?
Sure. Mitosis and meiosis?
Mm-hmm.
Okay, sure, we'll do. I'm just going to quickly go through the other questions as well, because it's a long answer. Lukman, question?
I just wanted to remind you about that question we were talking about in.
Oh, right.
Yeah.
Thank you. good memory. Let's see, where is it? Chat. Okay, okay, we'll go through a question shortly. It's a question for Ashida Lukman and Jessica. Question?
Yeah, I had a question. Is there like a disorder where people don't have antibodies and antigens?
Sorry, could you repeat that? You cut off a
Is
little
there
bit.
like a disorder where people don't have antigens and antibodies? And if
There are
so,
disorders where people don't have antibodies. Remember, people have antigens.
yeah, but
If you have molecules, you have antigens. So anyone that's made out of matter has antigens. The issue is some people can't produce antibodies. Very interesting conditions. You can search some of them up. You heard of if you search severe combined immunodeficiency, so SCID,
okay.
is the name of this disease. These children have to pretty much live in bubbles, literal bubbles. There's been a movie made about this because they don't have an immune response. They don't have functional immune cells and antibodies. And then there's conditions like A-gamma globulinemia, which is genetic. A meaning no, which means you lack it, and globulins is another name for proteins, specifically antibodies. So these people lack antibodies as well. There's a lot of interesting immune conditions as well. But yeah, good question. The simple answer is yes. Ramif, question?
Yeah, so I was doing this multiple choice thing, and there was this question, right? And it was like, which component of ELISA is responsible for the ability to detect an antigen at low concentration?
Sorry, did you say which property of ELISA? Is that what
Yeah,
you mentioned?
sorry, is responsible for the ability to detect an antigen at low concentration.
Wow. First of all, I didn't know you had to know about ELISA. It's a lab technique.
Yeah.
Do you know what ELISA is?
It's a way like viral samples are analyzed, right?
Yeah, ELISA is a fancy acronym for enzyme-linked immunosorbent assay.
Yeah.
What ELISA stands for. But pretty much what happens is if we're trying to detect an antigen, we will produce an antibody that's looking for that antigen. So
Yeah.
build a specific antibody, and we'll attach that antibody to a fluorescent dye.
Okay.
And if the antibody binds to the antigen, it will fluoresce, and that will give you a positive result for that antigen being present.
Okay, right.
So that's the basic. It's a bit more complicated than that. There's also sugar molecules, I believe, but that's just ELISA. Was this a short answer, an MCQ?
This was a practice question in my school, practice test, and I was mad because I got that wrong.
It was a practice test question.
Yeah.
Hey, look, it's out of the syllabus. Which is fine. Because you
Is
probably
it?
never learned it before.
Yeah, I don't think I've studied it before. That's why I was like, damn, that's a bit hard.
It's kind of out because you don't need to know this like all the different technologies. But
yeah, but it did give a bit of a blurb at the start. It was like is a technique that can be used to detect antigens in the first step, and like it gives a bit of a blurb.
Oh, then you should. Maybe send me the question, and I can explain to you the rationale behind it. But for everyone else listening, you don't need to know anything about ELISA.
Yep.
You're not given a blurb or information about it.
All right. Thanks, sir.
No worries. Tashfir, question?
Yeah, this is from last lesson, but with tuberculosis, I saw that there's this other type, or I think it's called latent tuberculosis. How does that work? Because it like activates later, but
Yeah, tuberculosis is weird. So what it does, it goes and infects your macrophages, which are a specific kind of white blood cell, and it can sit in these white blood cells for long periods of time. And what can happen is that TB activates this immune response, which causes a closed-off lesion in the lung. So this is a lesion which is walled off from the surroundings. It's kind of like I have some analogies for it, but I don't think we'll understand it. But it's kind of like walling off the bacteria, so it sits in hibernation waiting. And this TB can wait until a person is immunocompromised again. So if they're stressed out, if they haven't been eating or sleeping well, the immune system is weaker, and then this TB that's sitting inside this lesion in the lung will reactivate, redivide, reproliferate when white cells are a bit lower, and that can cause a secondary tuberculosis infection. We see that commonly in individuals so refugees, because they may have TB from their home country, and the travel conditions can make them immunocompromised as they're sleepless, they're prone to infections, poor hygiene. TB reactivates and they die when they land here, which is quite sad.
Yeah, I have this cousin who, like, it reactivated for him because he got the COVID vaccine.
Oh, yes.
Yeah.
Very interesting. Yeah. But yeah, what that meant is he had TB, was sitting walled off inside the lungs. It was quiet, it wasn't dividing, so he had no symptoms. And once he had the COVID vaccine, he would have been mounting immune response, or the immune response might have been weakened for a small period of time. And that led to reactivation of TB.
Okay, thank you.
Remember the question?
Yeah, so this is another random one, but I had a mate, right, and he had the vaccine and he had like myocarditis, like stage, like pretty severe one. Like, do you believe it's linked to that?
Vaccine is definitely linked to myocarditis, yeah, but it's kind of like it's the greater good, right? Myocarditis is self-resolving; they're not going to get any permanent long-term.
Yeah, he's 100% fine now, but
yeah. So even in severe myocarditis, like we typically just give steroids and pain relief, and they
right.
Versus getting COVID, which can cause long COVID disease, which, COVID's shown to drop people's IQ by up to 10 points. It's a multi-organ disease. But the simple answer is yes, and it's because some antigens on the heart look like COVID,
Right.
so your immune response attacks the heart, causing myocarditis as well.
It's inflammation of the myocardium, right?
The muscle layer of the heart. Yeah, it's quite an interesting disease. All right, I'm just going to call everyone back in. Let's jump back into things. So, Harshita, for your answering meiosis, I might do it after class. So please remind me and stay back, okay? Because we might not have time right now.
Okay, sir.
Is everyone inside from outside? Yeah, we got it right. Okay, cool. Now, quick question that I'm going to ask you all. Quick quiz, just so I know you understand everything. I'm going to go around asking questions, okay? So, we'll actually start with some of the online students. Brian, when it comes down to a plant, what are the two lines of defense that a plant has?
Passive and active.
Okay. Good. The way I want us to think about it is first line of defense is physical and chemical barriers, right? And the second line of defense is your active immune response. It's being kind of elicited or created. Very good. Pranav, what's the example of the plant and the pathogen that infects the plant?
There's a eucalyptus.
Yeah.
There's a fungi, which affects.
Good. Picinia, right? Picinia fungi causing wheat rust or eucalyptus rust. You can use either, honestly, as long as you have a plant. Good. Now, tell me an example of some of the physical barriers we have. So let me ask your turn. Good. One more. Good. Margin. Moving on. What are the active responses that a plant can engage in once it's infected? Good. Fungus command is one of them. Ethan, what's another one? Try not to look down. If you look down the nearest reading, right, then it becomes a kindergarten reading class. I want us to think of it from our brain. Right now, all this information is sitting in a hippocampus. It's not stored in your brain yet. The more times you elicit it without prompts, the more likely it is to move to your actual brain for memory. That's how learning works. Ethan?
Oxidative response.
Yeah, so try dumbing it down. If you don't want to use oxidative, which I don't like, I just say it produces free radicals with group electrons off pathogens. That's one, okay? And one more, Sashida? Yeah, so we've gone through cell walls. Yeah, good. The cell wall is the last one. And the final thing I will say is in severe infections, and we also do this, everyone, so it's quite interesting, we suicide our own cells. That is an actual response. Apoptosis is also known as programmed cell death. This is not forced. It's kind of like a samurai. Have you guys seen the samurai movies where if they get captured, they have themselves, right? It's kind of like that. That's what apoptosis is. Your cells and plant cells, when they become overwhelmed with a severe infection, they realize if they kill the cell that's infected, they kill the pathogen that's inside the cell as well. And that limits the further spread of infection. It's the greater good being done here, right? So your cells, quite ruthless, will just die off themselves. That is apoptosis. And that is a final response that even our body has in severe infections as well. Okay? Do we have any questions about apoptosis? Okay, you do need to know the word apoptosis. This is a word you don't need to know. I didn't understand it until medicine, but it's the exact opposite of apoptosis. Necrosis is when a cell dies because it's forcefully killed off by a pathogen, right? So whatever you've been thinking about pathogens killing cells is all necrosis. The only reason I'm labeling this is so you know the difference between this and programmed cell death, which is when a cell prepares to die. It does it very organized and peacefully. It starts breaking down its nucleus. It starts forming blebs that form off the cell, and the cell breaks down itself. Good. Forget about the delayed response. All of that's, and forget about extra examples. You don't need to know any of those. Good. Before we move on to the next dot point, I want to see if we can do some trial questions and see how we go for this. Have we already done this question? Very question. All right. Did we not do this for TB and HIV? Okay, sure. Well, you have 1 minute to jot down. Do you guys have any additional questions for them? You never got it? Give me a sec. Let me see. Let me go. In the meantime, do this question. Okay? You have a minute to start jotting things down.
What's the quote again? What? Malaria is what? That's 7 food. Yeah, please that and read it out. I close It's called bird virus. You forgot the viral RNA. Is your case study?
Okay, so let's go through this. We'll start with an example. You should have one go-to example for one disease. So, Ryan, what's your example of a disease that you can run through regarding all these categories? What example do you have? I think your internet might be a bit off. All right, let's move on to Victoria. What disease example are you going to use? Okay, we'll move on to Safia. Oh, Victoria, you've got an answer?
Yeah, sorry, my internet wasn't working. I was saying malaria.
Malaria? Okay, sure. Okay, pick one and stick to it. So, malaria, name the disease. What's the cause? Kushi? Is that the cause? It's a hard question. What is the cause of the disease? Plasmodium protozoan, right? Kushi, what were you talking about? Transmission. Yeah. So you should know transmission. They haven't included it here, but we'll add transmission. What would you write for transmission of malaria?
Vectors.
Very good. Vector. Female Anopheles mosquito. Good. This is a really good one to know because you're doing more with less. What I mean by this is you need to know about malaria anyway. You might as well just remember Plasmodium. Whereas I picked HIV, which meant I had to know a bit more. Good. What's the effect on the host? This is where you kind of need to know symptoms. You might have remembered when I was explaining what malaria does. Now you know why they ask that. Yes.
Like yellowing of the skin and the eye.
Why is that? Very good. Very good, right? So the protozoa can go and sit in the liver, and that can impair the function of the liver. One of the things that the liver does is it produces bilirubin. Has anyone heard of that fancy word before? So when your red blood cells die, one of the breakdown products is bilirubin, and the liver packages that in the gallbladder as part of bile. Does everyone know what the gallbladder is? No, it sits here. Your liver sits here in your right upper quadrant, and the gallbladder sits behind the liver, and it stores the bile, right? And it will release the bile every time you eat a fatty meal. So next time you go to McDonald's, just remember your bile is being released from your gallbladder, okay? But key point here, liver handles this waste product, and it will build up if the liver is damaged. And that makes your body look yellow. So yellowish discoloration, so I'd say jaundice is the word I'd like you to use. Jaundice. That's due to liver impairment. Right? Good. You don't need to know how jaundice works. That's way outside Okay, right here. Ria, what else happens in malaria? Pardon? Fever, definitely. It's a severe infection. Fever is one of the ways people die. Severe fever that causes organ failure as well. What's another effect? Yeah, you can get weight loss, all these nonspecific signs. Look, even if you said these two, you'll get the marks, right? They're not. You don't need to. You're not doing medicine here, but you're doing very good. A few other logical things to understand is malaria infects red blood cells as well. That's the main two sites malaria infects is red blood cells and the liver. When it affects the liver, it causes fevers, jaundice, all those things. It causes fever as well because it's present in the blood. Whenever you have bacteria in the blood, you'll get fever. Okay. The other thing is that it will damage and burst red cells. And when it damages and bursts red cells, what's inside red cells? What protein? Hemoglobin. Hemoglobin is toxic to the kidneys. You start getting kidney damage as well. So you get blood in the urine or dark colored urine as well. So those are other symptoms. As long as you understand, those are some symptoms of malaria. Let's move on. What's the treatment? Anyone know? This one's hard. Pardon? There's no vaccine for malaria.
I don't know about treatment, maybe prevention.
Good. What's some prevention you know? Some logical stuff, yeah.
Mosquito.
Yeah, repellent. What else? I've been wondering, well, it's so obvious, but that's as important as knowing the treatment, right? What are some other things you tell? Imagine you're a doctor. Someone's come to you, they're going to Africa or South Asia where there's high incidence of malaria. What else would you tell them? Clothing, right? So, very good. Clothing, full sleeve. Good. Okay.
I'm just going to quickly take attendance. Is Alyssa here?
Alyssa is here.
Okay. Georgina? Ashu?
Marshall's not.
Jessica?
Yeah?
Ian?
Not here.
Raya?
Is here.
Rhea? Tanisha?
Tanisha is not here. Tashfi is here.
Isha? Abhishek?
Yeah.
Kushi? Here. Sachita? Here. Lukman?
Lukman's here.
Kirtana? Here. Rashida? Gayatri?
Oh, sorry, Hashid is also here, and Gayatri is also here. Ramit's here.
Victoria?
Victoria's here.
And Savya.
Safi is also here.
I'm just going to invite
Do we have questions, Ramit?
Yeah, so have you heard of quinine?
Yeah. Okay. It's quinine. Okay, so what Ramit is talking about is actually how do I
Back in the day.
Indigenous Americans, they used tree bark and rubbed it on their skin to protect themselves against mosquitoes and malaria, and it worked. And we isolated the ingredient in that tree bark, and the name of that ingredient was quinine. We use an altered or modified form of quinine that goes by the name hydroxychloroquine. Hydroxychloroquine. Interesting, weird point. This is what you learn as well. A lot of medicine is actually very archaic. We just look at the traditions of what humans have been doing, and we simply try to isolate the raw ingredient, and we run clinical trials to find the best dosage. So hydroxychloroquine is one. Anyone knows Selena Gomez and lupus? This is another treatment for lupus as well. That's just a aside. Good. What are some more prevention methods, though? Lukman?
Using like mosquito nets for when you're sleeping. Or
Very good. Mosquito nets. Now, there's one last thing which I don't think many of you would know, but where do mosquitoes reproduce?
in water.
Still water. So what we can do is drain stagnant water sources. That is another population prevention method. Does that make sense? Really good. Yeah. I just Yeah, you can say hydroxychloroquine, but it's a long, fancy name, and I'm quite sure. Just know this. We actually don't use hydroxychloroquine much anymore because malaria is now resistant to that. So we use this triple therapy using different anti-protozoal medications. So you can maybe just say other anti-protozoals. Okay. How does a pathogen enter the body? How would you describe that? Yeah. Yeah, so it enters through an arthropod vector, which injects the Plasmodium protozoan where? Into the? Good. So it's injected by vector into bloodstream. Good. I'd highly recommend you all start doing these trial questions. I know you don't have a physical copy of you right now, but what we can do is it should be on the website, right? You all have access to the website, right? Hop on, start doing these questions.
Excuse me, sir.
Yeah?
I just want to let you know it isn't in the website. Also, it's not on the website.
Oh, it's not? Okay, thank you for letting me know. Well, I'll put the trial question booklet up. Okay. There's no rush. You guys haven't sat your Mod 6 exam yet, so be working towards that. Do you all remember the date you'll be sitting it? Yeah, so 21st, the Saturday, which is, I think, the 21st of May. We'll be sitting it there. Okay. Yeah?
Should I be sitting the Mod 6 exam as well?
I would recommend all of you sit it, right? And the reason is, even if you've recently joined, it's a really good way to assess where you are in biology and where you should be. Okay? So if you have learnt it already at school, I would highly recommend it. If you've never learnt Mod 6 before, you haven't learnt it with me either, then let me know. Okay? But most of you will not be under that case. Cool. Let's move on to this dot point. Well, it's quite a nasty image, right? Speaking of which, right now what I'm doing for my final year exams is we have hundreds of cases from all the different specialties in medicine to go through. And one of the cases that I went through the other day was a flesh-eating bacterial infection. And it looks a bit like this, but if you don't search this up, but this is a name of the flesh-eating bacterial infection that you can get. Necrotizing fasciitis. And if you Google search it, it looks really bad because you start seeing like this cutaneous gangrene, which is what you see here. This is dead cells, right? Dead cells on the surface start to become blackened. But you start to see the skin breaking down as if the bacteria is eating the tissue. So that's quite weird, but it does happen. Cool. Anyway, coming back to this, I want us to have a read of the dot point, and I want someone to dumb it down for me. I will get. Alyssa. Actually, Alyssa, I'm pretty sure your mic doesn't work, right? Ryan, could you dumb down this dot point for me? What does this dot point mean? There we go. While she's digesting it, I'm going to pick someone else as well. Ryan, is your mic working? Okay, we'll move on. Okay, we've got Tashia. What does dot point mean?
How animals can physically or chemically respond.
Good, right? Physical, chemical responses to pathogens, which cause disease. So physical and chemical responses to disease. That's what I want you to write down under that dot point. This is too wordy. Physical and chemical responses to disease. Very good. All right. Now, we'll start by looking here. You know, let's say you get into medical school and you go into first year of medicine, and this is the first patient you ever see. Any idea what this patient has? It's fine if you don't need to. But what do you think is going on using your understanding of Year 12 biology? Good. You see redness. You see swelling. Good. Georgina, anything you wanted to add? Good, right? So Georgina is relating this to what she understands. Something's causing the blood vessels in this person's lower limb to dilate, right? Now let's try breaking down what's causing what. What you're seeing here, everyone, is one of the biggest or most important responses to a disease that human tissues will exhibit. And this is the inflammatory response. This is part of the immune response, inflammation. You see this in every single condition, from malaria, where you have inflammation of the liver, all the way to this. This is an example of cellulitis, which is infection of the soft tissue, so the skin and the underlying fat underneath the skin. Okay, it's typically bacterial in cause. Any ideas why someone's put a marker and drawn around this person's leg? Pardon? Oh, yeah, no, not really. You don't amputate a cellulitis. You just treat it with antibodies. No, this person's not getting an amputation. It's to trap the spread of infection, right? See if it's spreading day by day. It's very some logical things we do in medicine, right? Cool. Now, when it comes to inflammation, two things happen at the level of the blood vessel, right? We spoke about this. The blood vessel goes from becoming narrow to becoming engorged, right? So one key process of inflammation is vasodilation. Okay, blood vessel becomes larger. What is another effect of inflammation? So one key component of the inflammatory response is vasodilation. What else did we say, everyone?
Permeability.
Yeah, so what also happens is the space between the cells that line the blood vessel also widen. And so that's increased vascular permeability. Now tell me, what signs would that cause? So what signs would you notice in vasodilation? Going back to this patient, which part of this patient's presentation is actually the vasodilation at play? Think about this. If you have more blood flow to a tissue, would there be a lump, or would it just be red and hot? Red and hot. If there's a lump, that's telling you something's moved into the tissue, which is not the vasodilation, but it's the permeability, which has allowed fluid to leak out of blood into your tissue. Does that make sense? So the increased vascular permeability is what's causing the swelling in tissue. Okay? Whereas vasodilation, it's carrying more blood to that area, so it's going to cause redness, heat. Blood is warm, right? 37.5 degrees versus your room temperature, so it's redness and heat. Now, when swelling occurs, it stretches nerve fibers in the area. What symptom would you have? Ouch. Pain, right? Very logical questions. Increased vascular permeability causes pain. So what you see here are the features of inflammation. Inflammation is the most important response that occurs in tissues in response to a pathogen. Okay, you can't miss that. Does that make sense? So now you understand what inflammation is. And my question to you is, why is this happening? Why would the body want to cause you redness, heat, swelling, pain, loss of function, which is the last part of inflammation? What's the purpose of this?
To increase fluid flow to the area, to deliver our white blood cells.
Very good. The number one goal, you cannot forget this. So the goal of the inflammatory response is to deliver. This is literally MCQ question 101. I've seen this in trial papers, and you'll definitely see it when you do Mod 7 trial questions, is to deliver white blood cells, which are in the blood, to move out of the blood into the site of injury. Does that make sense? To deliver white blood cells to site of injury. Very good. That is the goal of the inflammatory response. Does that make sense to everyone? That is why you get redness, pain, heat, all of those bad symptoms. It's because all that bad is outweighed by the good of delivering white cells that will clear the infection and resolve things. Okay? So next time you see these symptoms, it's a good sign that your immune response is working. Okay? Good. Do you have any questions so far? Okay, just the way you see this is the white cells are sitting in the blood vessel, and they'll move out, they'll move to the site of infection to kill whatever bacteria or pathogen is present. Good. I say that's the key thing you'll need to know, everyone. So that's the key physical change. What about chemical change? What would you all say? Chemical changes.
Hey sir.
Yep.
Sorry, just a quick question. Would you ever perform like a fasciotomy on a person with like that kind of inflammation?
Oh, very good question, right? Does anyone know what a fasciotomy is? So fascia refers to connective tissue in the body, right? So you might be wondering what's connective tissue. Your joints, your ligaments, those are all connective tissue. Fascia is connective tissue that surrounds your muscles, etc. Right? Now, the only time you do a fasciotomy, which is literally, this is what you do. And my friend's brother had this done to him a couple of days ago. You get a scalpel, you put them in theatre, and you make an open cut here, and you make an open cut on the other side of the leg, and an open cut in the bottom of the leg. They'll literally cut open the leg. Why would we do that? Don't go to hospital. The answer is, if you have what's called compartment syndrome, where let's say there's a fracture or there's a bleed in the leg, and there's so much pressure in the leg that blood is not getting to the leg, right? What would that mean? If there's so much pressure due to swelling or infection that blood is not getting to the leg, what will happen to the leg very shortly? No blood starts to die, and they'll need to get it amputated. So this is what we do to relieve the pressure in that compartment so that the leg can be salvaged. So the simple answer is no, you would not fasciotomy this patient, but you would if they had the other condition we spoke about, which is compartment syndrome. Good. Very interesting questions.
Thanks, sir.
No worries. Any other questions that we have?
Yeah, actually. Do you know how for increased permeability of the blood? Why would that result in fluid leaving?
So it's not increased permeability of the blood, it's increased permeability of the blood vessel, right? Think about this. Imagine this entire space is a tissue. Let's say this is a person's leg. This is one major artery that is traveling in the leg. When the artery increases its permeability, that allows plasma, or the watery component of blood, to also move out with white blood cells, right? And that's about 50% of blood is just plasma. And that's going to cause swelling of the leg. Does that make sense? Fluid will move out, the plasma will move out and cause swelling. Good. Now, the final thing I will add is, well, what causes this vasodilation and vascular permeability? You'll understand why inflammation happens, the purpose of it. You understand what is occurring in the inflammatory response with respect to blood vessels. My question to you is, how does the body know to inflame it? How does it know to make a blood vessel big? How does it know when to make it permeable? The simple answer is, if you think about pathogens, what do they like to do? They invade the tissue, right? Your tissue has these little bean bags of molecule. Inside these bean bags is a chemical called histamine. Anyone heard of histamine before? No? You heard of antihistamines before? Who has hay fever? Yeah? Can you explain to the class what symptoms you have? Yeah, sneezing, itchy, stuffy nose that's a bit swollen, right? The reason her nose gets so swollen is her nose is releasing a lot of histamine. Histamine is a chemical that causes the vasodilation and the vascular permeability. And histamine exists in little sacs all throughout tissue. When bacteria or pathogens invade and destroy tissue and cause infection, they will open up these sacs that are going to release histamine. Very good. These sacs are actually cells. They're called mast cells. But you don't need to know that. We're going to take that word out. Okay, so just know we have histamine. Infections will damage tissue that will release histamine that sets off inflammation. Okay? Very good. You do need to know this. You do need to know histamine because that is your simple answer for what chemical changes are occurring in the host cells. The physical change is inflammation. The chemical change is that the pathogen will damage tissue, releasing histamine and other chemicals. Good. Very good. Let's move on. Final thing I will talk about is this word here, before we finish today, cytokines. Now, the way you need to think of cytokines is they're chemical messengers. Chemical messenger molecules, right? And specifically, they're proteins. You know how you and I are discussing with each other using sound waves? That's our medium of communication, right? Sound waves get picked up by your ear, converted to an electrical signal. You understand that with some sort of meaning. Your immune cells talk to each other by releasing chemicals called cytokines. So in the immune response, your white cells, all the cells in the body will talk to each other using these cytokines. Does that make sense? That's all you need to know. All right? And that is it for today. I think some of you had questions. Stay back. I'll answer your questions. So Hashida, you look good. And Ryan, can I get you to stay back as well? Thank you. All right. Does anyone have any questions at all? All done? All right. Good. Guys did well today. Okay, everyone else, you're free to go. Okay, see you guys. Bye. Bye. Yep, no, I'll get you to stay back as well. Bye. See ya. Bye. Bye, Martin. See ya. Bye. See ya. Okay. All right. Narayani, question?
It's just a quick one. So, I don't have like this resource with me at the moment. So would I get it after like joining and stuff? Because
Yeah,
I
so
need to join.
good question. The way it will work is once you enroll, you can message admin to get a physical copy of this booklet mailed to you.
Right. Okay.
So this is my theory book for Module 7 that I've written up, right? It's got a couple of questions as well. It's pretty much everything you will need to know for Mod 7 plus more. So I'd say you may not even need to create your notes as long as you do more exam questions to compromise for that. And you should be fine. So yeah, so you just message admin and they'll get this printed and sent to you. The other thing is you should also log on to www.edxlearning.online.
Yeah.
Sign in via your Gmail account and then let admin know your Gmail so they can give you access to the exam questions, etc. in the website as well. Okay?
Right. Okay. I'll do
The
that.
last thing, Narayani, is since you're getting your notes mailed to you, you might as well also ask to get the Mod 7 exam booklet mailed to you. So this is all the past Mod 7 trial questions organized into one booklet for you to do to prepare you for your Mod 7 exam with me.
Okay, no worries. So would they also mail me the other modules as well, or is just module 7 at the moment?
They can. You just have to message them for that as well.
Okay, right. Okay, I'll do that. Thank you.
No worries. All right. Thanks. Have a good week. All right, Ramit, question?
Yeah, sorry, so this is a random question, but you said it's your last year of med school, right?
Yeah.
So if you could specialize into any faculty, what would it be?
Good question. I'd say surgery.
Right.
Only because I like doing things that are very hands-on, and surgery requires a lot of skill and expertise. You'll never get bored any day in your career. And it's a lot of quick dopamine as well, because you have to go in, you have to make quick impact and change. You see your patients improve almost instantaneously afterwards as well. And you're doing one of the scariest things. Like every day your job is different, and the worst thing that can happen in your job is the patient can die. So
yeah, that's correct.
You always need to be on your A game. So I like that responsibility, the challenge, the leadership, the science, the teamwork, and everything involved in it.
Yeah, that's great. All right, thanks, sir.
No worries. See ya. Lukman, question?
I just wanted to go through that question I sent you.
Yeah, sure. All right. And Ashina will go through mitosis, meiosis. Ashina, have you watched the prior lesson recording on mitosis, meiosis? If you go back to recordings, there's an entire lesson where we went through mitosis, meiosis. Have you gone through that?
I don't think so, sir, because that was part of mod 5 and I wasn't here for that half of the module.
But that's a good thing, right? Even though you weren't here for mod 5, if you go scroll up to the recordings for this lesson, not this lesson, sorry, on our team's channel, so you go to files, there'll be a folder called recordings, and you watch that, so go straight up to the top. That has everything explained for mitosis and meiosis. I'll still go through it with you. The only thing is, if you don't know any of that, and I go through it now, it's going to take a long time, and you won't be able to get through everything. But I'd recommend going through that after I explain it to you. Lukman, so your question was, do you mind if I share your chat channel, Lukman? Is that okay?
Yeah, that's okay.
Okay, cool. So your question was, recombinant DNA technology, how many genetically modified organisms should I know extensively? Oh, good question. You should know golden rice as more of an ethical implication of biotech. So you don't need to know too much about the specific methodology. You should know BT cotton in terms of its methodology. I'd say those two are sufficient. And probably also know E. coli.
E. coli, yeah.
The transgenic E. coli that make insulin, because that's a medical application of a biotech.
Yep.
So you're knowing 3 different recombinant DNA tech for different use cases. So for the method, you know BT cotton. For the ethics, you'll know golden rice. For the medical use of biotech, one example could be human insulin, or you could even just use vaccines or even antibiotics, because most antibiotics come from fungi or other bacteria.
And also that question, that exam question.
Oh, yeah. Group of families, 5 pandas in different zoos. Oh, I should have gone through this one in class. Sorry, I thought it was a comment that was the main question. Pranav, if you catch this question, there are 5 pandas in different zoos. Each family has been isolated for generations. As cubs reach sexual maturity, a mating program aims to, all of that's not, don't worry about all of that. Okay, they're isolated for generations. And this is the mating program. Mating program aims to include the genetics of isolated families for generations. The mating program helps to include the genetics of the worldwide. Yeah, so these are 5 different families. The only thing I'm trying to establish here is this mating program between the different groups, or are they still doing isolated mating within each population?
So, with that first question, I did get that question right. Yeah, and it's just, I think, well, since I got it right, it's basically saying how will the mating program aims to mate those different isolated families.
All together like this, is that right?
Yeah. So it would lead to an increased gene flow.
Yeah, it's a vague question, but if that was the case, then you're getting immigration of individuals into different populations. So that's your gene flow. Now, how about genetic drift? Okay, it's a really good question. Lukman, what are your thoughts on this?
So for genetic drift, what I initially wrote is that it would have no effect because genetic drift is by natural causes. So they can't really calculate like what's going to happen, but apparently genetic drift would decrease, which I don't understand.
Okay, now you said genetic drift is due to natural causes, which is vague. What are the two different kinds of genetic drift?
So there's bottleneck effect and founders effect.
Okay, bottleneck and founders, right? When does the bottleneck effect occur?
That's like a natural catastrophe.
Good, right? We don't have that situation here because they're in zoos and they're being protected, right? So we can take bottleneck out. What about the founders effect? Now, when we isolated these 5 different panda populations from the original panda population, what has happened to genetic drift then?
So it's increased because genetic, yeah.
Yeah, very good. So initially when we separate and create 5 different founding populations from original global panda population, that causes increased genetic drift because it's quite random which pandas we put in the populations for these 5 different families. But what occurs is when you start mixing these different panda populations together, they become more representative of the original panda population.
Yeah, and hence decreases.
Yeah, so that would decrease the founders effect. That's how I would go about it. So whenever you're stuck, work from first principles. What is genetic drift? What are the different types? Which ones at play here? And how is it varying?
Okay, yeah, that makes so much sense. All right, thank you, sir.
It's a hard question, so I want to go through it in the group class next week as well. Good.
Thank you.
All right, have a good week.
You too.
All right. And Hashira, was it mitosis and meiosis?
Yes, sir.
All right.
So even if you just explain meiosis, I think it's okay because mitosis is pretty easy to understand.
Good. I'd say so. All right, let's talk about meiosis. For now, you can eat if you'd like. Maybe you can help us out. For now, what are the different phases of meiosis? Yeah.
C and then there's P.
Good. So there's interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and another round of PMAT, which is prophase all the way to telophase, and a second round of cytokinesis as well. Okay. The key things I would say are with the first round of prophase, metaphase, and anaphase. So if I was to draw a cell for you, let's imagine that it has 4 chromosomes. Okay, Hashira? 1, 2, 3, 4. What happens in prophase, Hashira? Can you hear me?
Yes, sir, I can. I was just not sure.
Okay, no worries. So prophase is when the chromosomes will replicate, have a duplicated version of themselves, like this. Okay? They'll have that X shape that you've seen. Okay? But what also happens is we get crossing over, which is where a little piece, maybe I should have drawn the chromosomes in different colors. That's one homologous pair, and we'll do this as another homologous pair. Okay? What will happen is the chromosomes will exchange a little piece of each other's information. Can you see that being done here? The chromosomes swapping with the blue because they're part of one homologous pair.
Yeah.
And the green is going to be swapping with the orange, which is part of another homologous pair. What happens in metaphase, Hashira?
All of the chromosomes line up in the middle.
Good. Now, what happens is in metaphase of meiosis, so in mitosis, they all line up like this. They all line up vertically in one line, right? But in meiosis, they don't line up like that. In meiosis, they line up to the left and to the right, like this. Okay? And the key gist is that it's random. Let me label these chromosomes so it helps it out for you. 1, 2, 3, or 4. It's completely random as to whether 1 or 2 occupies its first position on the left. Okay?
Yeah.
But let's say that chromosome 1, as you see here, is occupied the first position on the left. Where must chromosome 2 be?
Opposite it?
Exactly opposite it, right? It can't be on the same side. And so now chromosome 2 has to be on the right if chromosome 1 is the one on the left. So what you'll notice is it's random as to whether 1 or 2 is on the left or the right, but they must be on opposite sides of each other. Do you see that?
Yes, sir.
That's called random segregation. Random segregation. It occurs in metaphase, but you also see textbooks say anaphase, so both are correct. But technically they've started segregating metaphase, anaphase. Okay? So what random segregation is that chromosomes within one homologous pair, so similar size and shape, move to opposite ends of the cell. Do you see that being done there?
Yeah.
Good. And then anaphase will actually segregate. So that's the segregation process. What also happens is independent assortment. Now, small tip with your exam, Hashida, in class, if you were to model random segregation, you should show me this arrangement. You should also redraw this with chromosome 2 on the left, chromosome 1 on the right, and then 3 and 4 the same. Because do you see what that's showing you, Hashida? It's showing you that chromosome 1 can be inherited with chromosome 3, or chromosome 2 can be inherited with chromosome 3. Can you see that?
Yes, so that makes sense.
Yeah, that's what's called independent assortment. When you model the two possibilities there of random segregation, you're modeling independent assortment. What independent assortment is telling you is that the inheritance of one homologous pair, so 1 and 2, is completely independent of another homologous pair, meaning they occur separate to one another, right? So you can inherit chromosome 2 with 3, or you can inherit chromosome 2 with chromosome 4, as you see in the case at the top.
Yeah.
That's independent assortment. So my tip is just when you draw your meiosis flowchart, make sure you show there's two possibilities of random segregation.
Okay, so.
Good. I guess to understand everything else, you need to watch the recording. Okay?
Okay, yeah.
All right, cool.
Okay.
Good luck.
Thanks, sir. Thank you.